And so most of the papers that I publish, I do an extensive review first before I publish the paper. And in this case, I found that over the last 8 years, I never been rejected in any journal because most of the paper, if you read my papers, I did substantive review for any topic that I ventured into. That means that all the questions for me from the reviewers and other scientists I have covered already when I write the paper. Okay, so this is a kind of trick that I realize that it's very helpful if you do not want to get rejected. Okay, so anyway, I found that there's about 106 species, but almost all of the species, or majority of the species, only one of them have been studied in one paper. So the most popular one is Cotyledoptera damicornis, but the rest of the other coral species have very few publications for them in terms of coral development. Okay, as I said earlier, coral sexuality is very complicated if we base it on our current understanding of literature. And I think even papers that are recently published still argue that there's a lot of effort needed to shed light on coral sexuality. So many corals are gonochoristic, but also some are hermaphroditic. But hermaphroditic can be split into the protogynous and protandrous, and even some corals are gonochoristic at let's say at the polyp level, but at the intercolony level they are hermaphroditic. So this kind of diversity of coral sexuality probably is a reflection of their very long evolutionary history. They have been here on this planet for over 500 million years since the Cambrian, and over that long period of time, they have experienced so many disturbances. So for them, maybe this variety of reproduction and sexual strategies enable them to survive many challenges. Okay, but for us, it's trying to have a really broad understanding in terms of variation across species, and if there's an ecological driver to this kind of variability, wherein species can express different kinds of sexuality in different environmental conditions. Because in terms of morphology, they can do that. The same species found in Apari can look like a flat, thin sheet. The same species, when you look in Central Luzon, is a branching coral that can grow really tall. So there's a lot of adaptation. Corals for me are like the perfect transformers. They can transform themselves depending on their environmental conditions, but there's not a lot of study in terms of the relationship of ecology and coral sexuality. So that's something that we need to investigate. Majority of these corals are actually broadcast spawners and brooding. Okay, so based on those that are studied, but sometimes there's actually a mixture. Corals that are broadcast spawners can also brood. So again, there's still a lot of area here to investigate. When we look at timing, initially this was my fear that I have limited time to study, and if the corals have seasonality like the ones in Great Barrier Reef, then that means that I'll miss some time because they will only spawn or release their gametes or their planula on in a certain season. But it turned out when I look at the literature that many different species actually have many different reproductive timing. So that means that probably if we just spend time out there in the ocean at night, then we might find some corals spawning at different times in the Philippines. But this is something that we need to establish. So I plan to maybe go every 2 months, say in Batangas, and do a regular observation of the spawning activities of corals, comparing across many different species. And the same with lunar control, and this is still also needs a lot of investigation because some people, scientists, kind of argue that it's not mainly the lunar cycle per se, but sometimes it's also affected by the solar radiation during that time for that lunar period. But if you look at this graph, actually corals also reproduce in various time around the moon. And in fact, some have no lunar period at all, and some continuously reproduce throughout the year. So I guess there's all this is saying is that there's really a lot of opportunity for us to investigate development of corals, and it's not because for me it's very expensive to do field work. You go to a place, sometimes you spend for 3 days to 5 days of something. covering your cost from renting of boats to renting of the place to stay and etc. Food to gasoline, etc. Your 50,000 could easily go away. And if you go there and you find nothing, you got no data, you're burning lots of money. So 1 million is very small money if you do a lab study at a large scale and comparing across species. So this is good news for me that I probably have more chance capturing on a given time and not wasting a lot of money. Okay, so in terms of egg size, there's a huge variation, but yeah, so some are very small and it can go up to 800 micrometer, and some are also only around 200, okay, and 100 in fact. Um, in terms of planula development, uh, again here there's still some kind of vagueness because although majority of those observations show that corals tend to be swimming after their full development, but it turned out that when they start settling, the corals actually have this tendency to initially have a phototactic. So many corals, planula, they already have picked up the symbiont at that stage, and they tend to be phototactic for some time because presumably the symbionts need the light. But at some point, because they have to settle, they have to actually turn off some of this phototactic and they start to become geotactic. So they attach, but they don't attach immediately. They kind of bounce back up and down until they probably find the most suitable um, environment. So I guess that the description of the activities of corals still needs further exploration of really whether they are simply crawling at all exclusively, or they do some crawling, or and then they do some swimming until they find the most suitable place for them to settle. Okay, and again, in terms of here the planula size, there's a huge variation, and here the settlement days. I think this is settlement days. Sorry, my is covering my. Why are you not going away? So hold on. I'll say settlement days. Many of the record that I look at, actually, some of them are very old publications. So there's one here that stood out 212, and that really kind of unbelievable for me, because how can one study and observe until 212 days? But I think majority of the papers I saw in a matter of week, corals already are fully developed and they start settling. Okay, hold on. Okay. So this I try just to show you the development pathways or patterns for the coral. This is just from one paper. I just to summarize, try to summarize what he found is that in initially the first 2 hours he already observed. So this is about 20 species of corals that he observed, and these are broadcast spawners. So eggs and sperms are broadcasted, and then they collected them and they observed the developing gamete and fertilized egg um, develop over 8 days period. And they found that there's a huge variation across species, but again, within half a day, 12 hours, there's already this development blastosphere. And in some species like Acropora, there's such a stage after the morula stage wherein the corals actually already form this kind of donut shape in preparation for further gastrulation. And pharynx and invagination of the oral pore happen within 24 hours, and the pharynx fully formed within 36 hours. So many are already mobile in about 3 days or so. Okay, so about 2 days at least. And by 3 days, many of these are already formed as a planula. Okay, but some already some kind of extend their development for a few more days, but majority of them are already settled by 6 days. Okay, now I want to also show some of the trends in terms of the study on the effects of various disturbances on coral development. Because this is part of the question, why why is it that when we look at many of these places, there's corals around us in this area, but there are no recruits. Even in places like Zambales, which is one of the oldest protected area in the Philippines, the other side of the island have a lot of corals, but the other side of the coral is degraded and there's no recruitment going on. So I think this can be related to some environmental factors that either prevent the coral from settling or surviving. And I look at the literature, but only few literature actually study the effect of environmental factors, even climate change, because climate change papers look at the bleaching of the adult corals, etc. Okay. Oh, I passed. It's only 246 already. But here, just to summarize, increased warming has been associated with a lot of stress for corals, increase in acidification, reduced mineral deposition, for example, a delayed metamorphosis, and reduced salinity, which is due to increase in rainfall, in La Nina period, for example, increase the mortality of gametes. So there's all these factors that potentially are preventing the coral larvae from surviving, even though the corals are reproducing and developing into successful cannula. So again, especially nutrients. So this is kind of related to our recent paper that all the highly sedimented protected areas have, in spite of the protection, have very low coral recovery. Okay. Sorry. So this is the gaps before I end my talk, and I think that there's a lot of opportunities that we can explore in the Philippines because we have these great resources, great diversity of corals. I think the question is really time and the patience to invest time underwater at night. I have done this when I was studying seahorse. It's not fun being cold at night underwater, staying there many hours, observing something, and sometimes you can get bitten by jellyfish or stung by jellyfish or bitten by a tiny damselfish. So, but there are no sharks. It's pretty safe in general. There's a large gap in terms of understanding of mechanisms and control for scleractinian coral development. So this is an area that needs to be further explored. Based on what I've seen in the literature, there's also a huge gap in terms of relating reproduction development with the entire life history, ecology, and phylogeny of corals. In fact, we do not have a good study of the phylogeny and of corals in the Philippines using, for example, molecular techniques. And there's also a great need to do more studies on the impacts of various disturbances on coral development and survival of larval cannula. Okay. So these are the major source that I got the data from for comparative purposes. Many other literature, it's very difficult to compare the data, although they are somewhat related. But they just give me an insight, but I cannot really compare the data to the other papers. So thank you, and that's the end of my talk. I also thank the various funding agency for my national coral reef field work. Thank you.
Maraming salamat po sa inyong presentation, Dr. Jonathan Anticamara. And ang dami po niyo sinabi, nakakatawa. I mean, in terms of the studies being done, and of course yung gap din, yung kulang pa. Ang dami niyo nasabi na napag-aralan na. Ang dami niyo na po napag-aralan. Ngunit kita-kita pa rin na ang dami pang dapat pag-aralan. Ang dami pa nating hindi alam at ang dami pang dami pang idagdag na kaalaman when it comes to coral ecology. Siyempre, before we go to the conservation, kailangan maintindihan muna natin yung basics na yun. So, para po sa ating participants, if you want to pursue this line of study, you can contact Professor Anticamara and maybe help him out in filling in the gaps sa sa knowledge natin when it comes to corals and especially corals in the Philippines. Ayan po. Now we go to our Q&A portion and we have our PSDB treasurer, Ms. Alicia L.J. Pagulayan, and she will be our Q&A moderator for this session. Hi, ma'am.
Hello. Good afternoon, everybody. Thank you, Dr. Pavia and Dr. Anticamara, for talking about the exciting world and the interesting world of corals as far as their development is concerned. At this point, we will have our Q&A, and I will first read the questions directed to Dr. Pavia, and then the next questions, the next three questions. I will read three questions for Dr. Pavia, and then the next three questions for our for Dr. Atikamara. All right, Sir Pavia, this is a question. Do the eggs and sperm of a super coral produce a super coral, or do they revert back to individual coral strains?
Yes, with that regards, that's something that, like I said, needs to be studied. We have to remember that, a coral per se will produce the egg and sperm bundles, from its gonads. But of course, when we look at how a chimera performs, like I said, it could be easily said that, for example, if you have the component of, let's say, genotype A producing eggs and sperm from genotype A and then B and C, but in bryozoans, they've documented a sort of gonadal parasitism where there's no phenotypic expression of the other colony that was fused. But when they collected the eggs and sperm from what survived, like when A and B fused, B disappeared. But when the eggs were produced, it had the genotype of B. So B disappeared from the colony, but it persisted as a parasite in the gonads of A. So that will also be part of the greater study on how the chimera works, because we still don't know. I mean, there's very limited work on mature chimeric colonies and how they work, especially in small polyp corals where there's a lot of dynamics that need to be ascertained if ever, with a very stable chimera, if it is formed.
All right, thank you very much. The next question is there horizontal gene transfer in corals? For coral chimeras, could substitute corals exchange parts of their genome? This is also related to the first question.
When you look at the reproduction in corals, the way I see it for now, when we look at the chimeric corals, I'm simply looking at the possibility of creating a coral composed of numerous genotypes that are working as a single unit. The reproduction per se, we're not sure if it will conform with the usual reproduction of a single clonal colony, but that still remains to be answered as soon as we get mature reproducing chimeric corals.
So there's still really a lot to understand.
Yes.
In that field of producing chimeras. But the formation of chimeras is naturally observed.
Yes, it's a natural occurrence because when recruits from the same species settle near each other, the tendency is for them to fuse, to come into contact and fuse as long as they're the same species. And they've observed that the fusion is non-problematic, at least for the first 3 months, because the immune system has not developed yet. And observations tend to lean on the explanation that, because if getting bigger faster is the way for the colonies to survive better, then maybe that's the reason why the immune system is suppressed at that stage. And of course, if it occurs naturally, we have a lot of chimeric corals, but since they look very similar to each other, like a brown Pocillopora and another brown, Pocillopora, same species, it takes, for example, maybe using fluorescent dyes or using UV flashlights to really determine, what happens in the union. That is something that we have barely opened.
Okay, thank you. And the last question for Dr. Pavia for now. Are there cases on which the chimeric corals contribute to the declines of their parental species in your experiments?
There should really be no decline in the parent species because it's still the same species. and it's something that occurs normally in the wild. It's something that happens by chance. We're talking about the same species just coming together and fusing. For me, it leads to better genetic diversity for that species. It creates more robust organisms of that species, and we're not even talking. Although there have been whispers that maybe different species of within the Posilopora occurred. Like sometimes I see a small colony that one half looks like Posilopora, and the other looks like Eduxi or another species, but still within the same genus. I won't even go into that. But looking at what we have, what we're doing, what we're planning to do in the future, there should be no risk to the species because it's something that happens normally.
Okay, thank you, Dr. Pavia. The next questions are for Dr. Anticamara. The question is the literature about coral conservation accessible to fishermen communities? Do you have data on the open, closed status of the journals? Sir, please unmute.
Thanks for
Okay,
that
thank you.
question. So I have spent about 5 years before I started my PhD living in communities, fishing communities in Bohol. There are many, many islands in Bohol. There's a place called Double Barrier Reef, only one of two in the world. And in there's many, many fishers. And yeah, no, the education level in many of these communities are relatively low. Some even don't have high school in their villages. So we have with some of the people that I work with, we established some schools by asking for donations from companies to establish schools. But anyway, so reading is very not it's not something that they will do. Reading definitely scientific journal, and so the way we make it as accessible is by doing some kind of environmental education program, wherein we simplify information from the literature and present it to the community. So that's how we increase the number of protected areas in Bohol. Otherwise, when I started in 1997, there's only one protected area, and my task was to establish more. And most people are against protected area because that will take away their livelihood. So I have to do a lot of this environmental education, but the kind of environmental education is not really just lecture. You have to bring them to the ocean, show them. So I think sometimes the scientific literature is not that relevant. It's picking up some information, but also applying that to something in their context that are more useful for them and they can relate to. Otherwise, if it's just a scientific paper, I think they will just be bored and they will not find it very relevant. So yeah, so there's a lot of responsibility there for conservationist scientists to reach out to these communities and simplify the very technical scientific literature and relate it to the lives and livelihoods and situations of the people in fishing communities so that they can solve some of their problems.
Yeah, thank you. The next question is, could we be missing important coral work by low and middle income countries like ours by focusing on journals indexed by Scopus? How do we address this?
So that's why the way I address that is that when I do extensive literature review, I start with Web of Science or Scopus because that's the standard. Whenever you read review literature right now, usually everybody does that. But as I said earlier, when you do that, you're missing some. So what I do is I add more searches using Google Scholar and etc. and other search engine. And I after I do my extensive review from Scopus or Web of Science, I then look at this possible other literature that I missed. So the thing is that it's not there's a difference between missing and also there's a difference between the fact that there's none, there's very limited. So if we search and search, even how long we search for, let's say, estimate of diversity of corals in the Philippines, there's not a lot of research and there's not a lot of scientists working on corals in the Philippines. Very limited number of people. and many of them do not work on all many species, so very limited species as well. So I think that to some extent when we search in Scopus or in Web of Science and we don't see anything, to some extent that's also a reflection of the fact that there's very limited um,
Right.
literature. And as mentioned earlier, I've been working for 10 years. I've been working with UN, and you know the UN, for example, the UN IPCC report. When you read that report, it's really from a synthesis of all scientific literature. It's not an original research. And when they do that, they actually have to assess not only the availability of that literature, but also the quality. So I think sometimes it's not just the question of missing the literature, it's also the reliability of the findings, how good is the science in this paper. Although it's from a developing country, and if it's really poor quality, we have to also carefully judge or not judge, but evaluate the quality of the paper, so that at the end of the day, still our goal is to have the best science, and that because it's yeah, especially in this case, it's a large field. There's many people out there in the world studying corals. Science is advancing fast. It's just that it's not advancing as fast in the Philippines. So to answer your question, I'm trying my best to incorporate some from the Philippines, but at the end of the day, still very limited compared to the rest of the world, because nobody is spending enough money on coral reef research. It's hard to find money for coral reefs.
Thank you. And the next question is, with regards to restoration of corals, how long is this process?
Ah, so if you look at the available literature in the Philippines, again, it's limited. We do not have a long-term study on coral restoration. But if you look at the restoration of corals from across the globe, there's few long-term studies already. And in the Philippines, there are restoration projects. But so, example, one time I attended a Senate meeting on coral restoration, and I think the restoration, typically a project in the Philippines about 3 years, right? Funded for 3 years, and usually you don't get an extension for another year. You have to change a topic or you have a different part of the topic. So there's not really a long-term funding for most activities. So it's difficult for conservation work like restoration. So what happened in that Senate meeting, for example, I think some of the senators asked, so after you restore, what did you do? Did you monitor? But many answered, no, we did not come back. We did not monitor. I think if I publish the paper on the blocks, probably I think it's probably one of longer time monitoring of consistent of standardized block. But I think overall, my belief, based on what I observed, is that if we restore coral reef, given the dynamics of coral reef, it's not something that okay we can achieve in 2 years, 3 years, and then we solve the problem. For me, I think it's like managing a resource. There should be a committed group of people who will monitor, because we can't predict what will happen to that coral reef. So therefore, we need to develop adaptive management. If something happens, we will need to do X. If something happens, we need to do Y, something like that. It's going to be an ongoing process, because we need to ensure that the coral reefs can produce the services and the goods that the people need for people to survive and be healthy. So therefore, the management, the restoration is not something you do once and you hope it will solve the problems in 2 years. It's gonna be for as long as we live. We manage coral reef, we study them, so that along with the people, the reefs will survive, because the people are dependent on reefs. So yeah, so I don't think it's. I wish that people don't view restoration and management as something you do and then you stop doing. It's something that we do and then you continuously doing. Whatever the resource need in terms of management and restoration.
Thank you very much, Dr. Anticamara, for all your insights. We will go back to Dr. Pavia. Hello again, Dr. Pavia. The question here is, are there any possible problems on the reproduction of chimeras?
I mean, bottom line is, basing it on the fact that they're the same species and they're just clones of the same species, theoretically there should not be any problem with the reproduction. But of course, when you talk about the quality of reproduction, I mean, I'm just grasping at straws here because we don't have, or at least I have not studied any mature chimeric colonies yet. But what I'm looking at is if there's no serious immune response between clones, there are no clashes within the coral. It's just different genotypes of the same species living in one coral colony. There should not be any problems with reproduction. But again, the final answer will be later on when we try to look at the reproductive effort of if we ever have mature colonies and we start looking at diversity, we look at survivability, we look at all the other aspects. That's the only time when we will learn about these things.
Okay, thank you, Dr. Pavia. The next question is, what is the effect of global warming on coral development? I often hear coral bleaching, but is this applicable to adult corals only?
I mean, bleaching occurs across the board as long as a coral is, a coral with the symbiotic algae, as long as they're exposed to thermal stress, then that's the first thing that we observe, that because it's an acute event, temperatures rise, the coral is stressed, it's going to release the algae. But like I mentioned in the early part of my presentation, it goes more than that. Some of the more lingering effects are increased acidity of the oceans, which we're not at that tipping point yet, but we do know what is going to happen based on laboratory experiments. Calcification is affected two ways because calcium won't be available readily for use of the organism, so growth will be slowed down. And also for some coral species, like some of my students are working on it now, they're looking at some corals that live near CO2 vents, and they're trying to see, are the skeletons more brittle? Are they more porous? Because growth may not be affected, the growth rates may not be affected, but the quality of the calcification may be. Meaning a coral will grow at the same rate, but the skeleton may be riddled with holes, or it may be very porous, which means a single storm can come in and wipe out all the corals that have brittle skeletons. So those are some of the effects. So bleaching, skeleton formation, and also, of course, one of the more indirect effects would be like what are being predicted by climatologists: increased precipitation and a rise in sea levels will cause massive erosion along the seashore and inland outwards towards the rivers. So it will cause either a lessening of the salinity, a reduction of the salinity in near shore areas. But aside from that, there will be turbidity events, which would, of course, shade the corals, and the sediments settling on them will provide a negative impact. We've been observing this in other locations already, but not due to climate change, due to mining or other things. So these things will affect corals. It's not just the warming itself that affects them.
Okay, thank you very much, Dr. Pavia. The last two questions are directed to Dr. Anticamara. On regrowing fragmented corals, what are the species that show the most promising results in terms of repopulation?
Sorry, I have to go back to the paper, but it's mostly Acropora. So it's not one species, but it's several species of Acropora. Because Acropora, the branching corals, they tend to grow faster. But they also are more susceptible to stress. So although they grow faster, once there is a typhoon, they break faster compared to other genera. So yeah, so that's the short, because I have to go back to each and individual species. And I studied several species, so I have to pull back the paper. But anyway, Acropora is the answer. Many of the genus for Acropora, many of the species under the genus Acropora has the potential to exhibit rapid recovery when reattached from a small fragment.
These are the Acropora are the ones that have thin branches and they readily break.
Yeah. So if I show you the slide again, if I share the slide, so this, if you can see this one, sorry.
Is that the Staghorn Coral?
Yeah, so some staghorn corals, but there are so many species of Acropora. I have a current work where I do their genetics so that I can because some of them actually have very confusing taxonomy. So right now I'm doing a molecular analysis so that I can finish the paper so that I'm certain that these are separate species. So sometimes there's also a challenge of the identity of the corals just by morphology alone. So that's why I'm kind of cautious about publishing all my coral papers until I finish that. But I finished some of the molecular analysis already. So I'm pretty certain about the species, relatively, because I only use a few markers. But still, that's good enough than just basing on morphology alone. Okay, but these are Acropora, and I should have. I have a paper in Science de Demand where I summarize all the growth rate of corals, and in fact that has been cited by international researchers because there's very few papers. There's several papers on restoration, but there's few standardized way to measure the growth rate. Because, for example, how do you measure this coral, this coral versus this coral versus this coral? So what I did is actually not to measure just the length, but the area. Because at least with the area, I can actually compare across the several species. So I measure the growth rate in terms of the area, not the length of the branch. So this is an Acropora. This is an Acropora. This is an Acropora. Okay. So yeah. Anyway.
Thank you. The last question is, what contributes to the variety of coral reproduction for Dr. Anticamara?
I think it's probably the same reason for the diversification of the coral. So for me, the coral reefs and the reef. So I think that the diversification is partly due to the long evolutionary history. So they had that long period of time wherein they have experienced so many stress and therefore they were selected. And then they were selected, therefore they diversified, speciate, something like that. So I think that the same reason for the diversity of reproduction. So when we see mutations, for example, it leads to some morphological, but sometimes it's also behavioral consequence. So we can say that, for example, reproduction is part of the behavioral strategies for corals, but it's also driven by their internal physiological. So I think overall, I have no short answer, but I think what we need to do in order to have a good answer for that question is really have a comparative analysis of the development strategy of corals, because they can, as I said earlier, they can change their strategy from one environment to another. So therefore, if we can figure that out, then maybe we can figure out what contribution does the environment play in terms of how corals decide whether it's going to be a hermaphrodite, it's going to be a broadcast spawner, or it's going to be a brooder, it's going to be a broadcast spawner. Is it going to be, because it's also a sequential hermaphrodite, so is it going to be a male this time or it's going to be a female? So some things like this we can figure out if we study many species of corals in the Philippines, and we have a better answer to this question.
Okay, thank you very much, Dr. Anticamara and Dr. Pavia, for graciously answering most of the questions that were uploaded in our Slido link. There are still several questions, but in the interest of time, we have to end our Q&A portion. So I will turn over the stage to Dr. Dino Tordesillas.
Maraming salamat po, Ma'am Alice. And thank you for leading our open forum. And once again, thank you to our speakers, Dr. Pavia and Dr. Anticamara, and for sharing important knowledge and insights on coral biodiversity, coral conservation, coral development, coral reproduction. And again, for our participants, if you are maybe you're leaning towards this field of marine biology, or any thing to do with corals, maybe you can reach out to our presenters or our speakers, and maybe you can come up with some collaboration or collaborative work in the future. Okay, so we would like to again thank our speakers for their for sharing their knowledge, and as a way of for us to show our appreciation, we present you these certificates of appreciation. So we have certificates of appreciation given to Dr. Richard Thomas B. Pavia and to Dr. Jonathan A. Anticamara for their generous sharing of knowledge and expertise as plenary speakers during Session 5 of the Philippine Society for Developmental Biology 13th Annual Scientific Conference, held online from November 17 to the 20th this year, 2021, signed by the Secretary, yours truly, and our PSDB President, Dr. Larry Ann Ipulan-Colet. Uli po, maraming salamat po, Drs. Anticamara and Dr. Pavia. Hopefully sa susunod po meron po tayong iba pang chance at ano po, face-to-face na po tayo, and hopefully may school na mag-host sa atin na pwede tayong makakita ng corals talaga in action. So in the future po, ano tayo, hopeful tayo na we have that opportunity once again. Maraming salamat po muli.
Thank you so much for having us.
Thank you.
Okay. So
Salamat po.
bye for now.
Okay. Before we proceed to the other parts of our the last part of our conference, not just the session, we would like to invite our participants and our speakers, okay, for this session to for a photo op before we end the session. So isang mabilis lang po na picture again. We invite Dr. Pavia and Anticamara ulit to turn on their cameras with us for a photo op. Saan nandiyan pa sila? Okay. So I do hope they're still there. Okay, so for session 5, kindly turn on your cameras. Picture po tayo ulit. So, hintayin natin, baka may mga hahabol pa po. Ayan. So, ayun, ito po. I think, ayan, may mga ano pa, mahabol pa po. Okay, so I think that's, we just have one panel for everyone. Ako na po kukuha. Okay, sige po, tingin po tayo sa camera with our big smile. 1, 2, 3, smile. Isa po, lagyan natin ng mga, medyo wacky naman tayo. 1, 2, 3, smile. Okay po, maraming salamat po to our attendees. At hindi pa po dito nagtatapos. We still have the announcement of the winners for our contests. Let's go first with the photo contest ba muna or poster?
I think poster muna, sir.
So I think we should go first with the photo muna po tayo. Let's go first to the photo contest. I think we have several finalists here. Okay. So may I ask may I ask Jarel to present our winner for to share the photo, our winning photo pala for this session, for session 5. I think we have 3 entries. And can I ask Jarel to share the winning photo so that other participants can
Okay
see
po, sir.
kung sino po yung ating nanalo?
Okay po.
Sige lang. Okay, so while waiting for the winner for our ayan, so ito na muna. Okay, maraming salamat, Jarel, our host for session 5. So our winner for this for session 5, session 4 kanina umaga, but this is session 5. So our winner is okay, medyo ano, it's Carl Yusef Pelayo, and saktong-sakto yung kanyang photo with corals, and his caption is, "Coral chimeras are important in ecological diversity. I learned that they serve as barriers to protect us against waves, floods, and storms." So congratulations, Carl Yosef Pelayo. We'll get in touch with you through the PSDB Secretariat will get in touch with you for your prize sa pagkapanalo mo as our in our photo PSDB photo contest for the 13th annual scientific conference. So maraming salamat and congratulations, Carl Joseph Pelayo. Okay, so thank you, Jarel, for that. And we go to the next winners, and this is on the poster presentation competition. And I would like to ask Sir Ahmad to present our winners. Sir?
Sure, with pleasure. Okay, sige. So, third place, okay, for our third place, ano natin, we have the poster of Maria Claret L. Tsuchiya and Kazushiko Tsuchiya entitled Inexpensive Research at Home. Slideshow muna. Slideshow naman natin. Okay. Observing the stages of egg development in pet Medica. Is Ma'am Tsuchiya here? Baka pwedeng paki-spotlight natin po siya. Ma'am Lalet, if you can please, sorry, didn't mean to do that. Open your cam. So, ayan. Okay, third place. Congratulations, Ma'am Lalet, Tsuchiya.
Thank you, PSDB.
Alright, sige po. Ayan, para naman, kasi wala tayong stage eh. For the second place, we have the poster of Miss Christine Nicole M. Padilla entitled Optimization of Protocol for the Transplantation of MCF7 Mammospheres on Duct Chorioallantoic Membrane. Is Miss Padilla here with us? Okay, sige. Okay, congratulations. Okay, yan. Kasi wala tayong stage eh. So, ano. And for the first place, okay, this went to Mr. Jobriel Baluyot et al. And the poster is entitled Detection of Anti-Androgenic Activities in Laguna Lake Surface Water Using Transcriptionally Activated Human Cell Line. So, Mr. Jobriel Baluyot, congratulations. You are the winner of this year's poster competition.
Thank you po, PSDB.
Okay. Sige, ayun po. Para po makita lang, para naging ano, so we had preliminary and Q&A round. Ito po yung naging scores natin. As you can see, may mga naging dikitan labanan, but our judges did a fantastic job delivering everything. So thank you very much. And we hope with this, we would encourage everyone that even in this time na talaga mahirap kumuha ng data, hindi pa rin tayo hindi ba? Mawala ng gana mag-present sa mga scientific meetings pa rin. So PSDB through the secretariat na lang will get in touch in order to give you your prizes. Yes, you have prizes for this aside from pero ang pinaka-prize dito is bragging rights. Okay. So, thank you very much. Ibabalik ko ulit sa ating host, si ating master si Dr. Dino Tordesillas.
Maraming salamat po, Dr. Ahmad Reza F. Mazaheri for that. And salamat po sa poster committee at ang head po nila, ayan, si Sir Ahmad. Aside from Sir Ahmad, I would also like to thank our judges. Okay, Sir Ryan De Chavez from UPLB, si Ma'am Joyce Ferrer from UP Diliman, and also Ma'am Sonia Jacinto for serving as our judges at helping us pick our winners for our poster presentation. Okay? Ngayon po ay we do have one more chance for people to win. We have a pa-quiz, may pa-quiz kami, okay? At pero instead na grade, ay prize po ang ating makukuha. Let me give the floor to Jarel who will serve as our quizmaster. Ikaw ba ang quizmaster natin ngayon? So, Jarel, go ahead and magpa-quiz ka na.
Okay po. Thank you po, Sir Dino. So, good afternoon, everyone. So, right now, we will begin with our game quiz. So, I am Jared. I will be your game master. So, we request everyone to participate for this quiz by clicking the Slido link posted in the chat box here in Slido. And so, here are the mechanics of the game. So, first, kindly put your full name as well as your institutional affiliation. So, kapag click nyo po ng Slido link, dun po sa may part ng dun sa poll, ayan, dun po sa poll. Ayan, pa-click na lang po and then pakisulat po ang inyong full name and yung inyong institutional affiliation. Okay, so as of now, we have 19 participants. Wala mong gustong sumali, malalaki ang ating prizes for the top scorers. Okay, so okay, so while waiting, so we would like to thank our sponsors, Macare Medicals, Merck, MedTest, and Phil Anasur. Okay, so right now 25 participants na ang nag-log in. sa slide ulit. Okay. So, okay, second instruction. So there will be 6 questions and each question has their own time limit. So you need to answer each question as soon as you can. So the questions were extracted from the speakers and also from the abstracts of the competing posters. And then third, top 3 winners with the high scores and the fastest will be awarded. Okay, so let's start na, Ma'am Larry. Okay, sige. Let's start na. First question. Rafflesia is easy to culture in vitro. True or false? Okay. Okay, second question. Okay, so 30% answered true and while 70% answered false. So ang tamang sagot po para po dyan sa question number 1 ay false. Okay, question number 2. What assay was performed to observe the effect of the Garcinia binucau crude leaf extract on myoblast count in 24 and 48-hour intervals? CAM assay, proliferation assay, immunoassay, or induction assay? Okay, time is up. So 17% answered CAM assay, 44% answered proliferation assay, 17% answered immunoassay, and then 22% answered induction assay. So the correct answer for that question is B, proliferation assay. Okay. Question number 3. Which of the following aquatic animals were considered to have the most diverse vaginal morphologies? Seals and fur seals, whales, dolphins, and porpoises, sea otters, or corals? Okay, time's up. So 6% answered seals and fur seals, 88% answered whales, dolphins, and porpoises, 6% for sea otters, and then 0 for the coral. So the correct answer is whales, dolphins, and porpoises. Okay, next question. Ayan, so nakikita niyo yung mga points. So ang nangunguna ay si Aljon Borabo ng DMMSU with 25 seconds, Robbie Tagalog from STI, and then Jason Bitakura from VSU. Ayan, okay. Next question. What specific gene reporter was utilized in the detection of anti-androgenic activities in Laguna Lake surface water? Okay. Time's up. So ang sumagot ng beta-galactosidase ay 22%, chloramphenicol is 22%, luciferase is 44%, then beta-glucuronidase is 11%. So, sige po. So, ang tamang sagot ay luciferase. Ayan. Sino na po ang nangunguna sa ating leaderboard po? Okay, sige. Before that, go na tayo dun sa question number 5. Which of the following is best designed to have the best possible genotypic and phenotypic qualities that can allow coral reefs a better chance of surviving expected climate change effects? Super colony, super water, super science, or Superman? May plus points po pag sumagot tayo ng Superman. Ayan. Okay. Time's up. So, everybody answered super colony. So, yan po ang tamang sagot. For the last question, okay, to what specific organ of the Danilo was the teratogenic effect of CPA or cyclophosphamide performed? Heart, liver, eyes, or kidney? I think this was one of the doon sa ating poster abstract. Okay, time's up. So, ang tamang sagot po for this one is heart. So, 72% po of the participants answered this question correctly. Okay, for the result. Okay, so ang nangunguna ay si Jillian Uy from UPD with 6/6. So, ang kanyang time is 1:07. We have Juris Justin Motatovera also from UPD Liman, so 6/6 points din. And then si Miss Ria Alcabedos na mayroon 6/6 points, and meron siyang time na 1 minute and 47 seconds. Okay, so that's it. Thank you. So the PSDB Secretariat will contact yung itong first three na ito na nanalo sa ating game. So thank you, Sir Dino. Take it over
Okay,
po.
maraming salamat, Jarrell. So just to clarify, top 3 yung kasi tayo sila, 'di ba? 6/6, pero dahil sila rin yung tatlong pinakamabibilis, sila rin yung nanalo. So muli po, congratulations. Salamat, Jarrell, sa iyong napakahirap na pa-quiz with matching pa-Superman na sagot. We have again our winners. Rika, all three of them got all the answers correct. Let me just share the results. So here, again, we have all three of them got 6 out of the 6 questions. Nagkatalo lang po sa pabilisan ng pagkasagot. So we have Ria Alcabedos with a time of 1 minute and 47 seconds. And then so she's on 3rd place. 2nd place is Juris Justin Mota Tovera with 1 minute and 13 seconds. And our grand winner sa pa-quiz po ni Sir Jarrell ay si Miss Jillian Uy with just 67 seconds for all the 6 questions. So parang 10 seconds each lang, parang ganun. A little bit more than 11 seconds. So maraming salamat and congratulations po sa ating winners ng ating pa-quiz. Ayan, so I think we are now at the very end of our not just the session but our conference. Let me just share. Okay, wait lang po. By the way, before we close the session, not just the session but the conference, we do have we need the members to stay because we still have business meeting after this. Wherein we will the president will report, the treasurer and I myself, the secretary, will give the reports for the events that are the important details that are necessary for our members to know. So and also since this is a members only event, the business meeting is only a members only event, and only members can get certificates. The evaluation link will be posted after the business meeting for those who are buying for certificates, right? And okay, so we are now at the last part. Our closing for our closing remarks, we have our PSDB Vice President, Ms. Nadia Palomar Abesamis from Silliman University. Ma'am, nakamute po. Yan.
Okay, take 2. Thank you, Dino, and good afternoon to everyone. It's really been a very enlightening past 4 days. No, the 13th PSDB Scientific Conference indeed showcased diversity in developmental biology. So we went from the life cycle of a crown of thorns starfish to the evolution of crown oomycete lineages, from the development and propagation of parasitic Rafflesia plants to the evolution and diversity of penises and vaginas in ducks, snakes, alpacas, and marine mammals, from immune cell niches and therapies for osteoporosis to the role of cytokines in the learning behavior of mice. And then finally, from creating climate change resilient coral chimeras and to the coral reproduction and development research and gaps, especially in the Philippines. Well, that's a lot actually, overwhelming yung information, but one can't help but marvel at how magnificent and mysterious the living world is, no, after these 4 days of learning. So at this point, I would like to thank all of our participants, PSDB members, and our sponsors who joined and supported this year's scientific conference, as well as the other activities organized by the Philippine Society for Developmental Biology this 2021. Despite the pandemic, no, and all of these virtual events, the PSDB tried its best to still provide venues for learning and sharing among researchers, teachers, and students working in the field of developmental biology. So I would like to close this year's scientific conference officially by wishing everyone continued good health, safety, and a pleasant weekend to all. Maraming salamat. And members, please stay for our business meeting. Thank you.
Maraming salamat po, Ma'am Naja, for delivering your closing remarks for to close this the 13th our 13th annual scientific conference, and with the theme diversity in developmental biology. And before we close, I would like once again to thank our sponsors. Okay, so let me just share this. Oh, sorry. So yun, let me just share our this. Again, the Philippine Society for Developmental Biology would like to thank MedTest, Medical Test Systems Incorporated, PhilAnaserve Incorporated, Merck, and this in our 13th annual scientific conference is in cooperation with Macare Medicals Incorporated. Muli po, maraming salamat po sa our sponsors, and of course, maraming salamat din po sa mga participants namin, not just for today, not just for this morning, but for the past the last 3 weeknights that we held the sessions 1 to 3. Maraming salamat po sa pagtangkilik at patuloy na pagsuporta sa Philippine Society for Developmental Biology. Ayun po. So I think that's it, not just for the session, but for the conference. We would again like to ask our members to please stay for the business meeting and for the post-conference evaluation. May mga nagtatanong po pala, may evaluation po daw ba for session 4? Wala po. So we have just one post-conference evaluation which will be given to the members attending the business meeting. Muli po, maraming salamat. I invite the board members to turn on their cameras and maybe we can show our appreciation to our participants. Ay, maraming salamat po sa lahat ng dumalo at nagtanong, nag-participate, nag-post ng photos, nagpa-picture. Salamat po sa inyong lahat. Maraming salamat po.
So let's just give around 2 minutes for all non-members to exit the Zoom meeting. And then all members, can you please stay? And we'll have our members